Hello, everyone. I'm Tiago Ferreira from the NIH, and today I'm going to talk about sex in Leishmania, a largely unknown process, and how we are using single-cell transcriptomics and other tools to study mating in these parasites. If you ever walk into any lab growing these parasites and ask to have a look at a parasite culture under the microscope, you will most likely see something like this, which is the extracellular motile life cycle stage, known as the promastigote. So Leishmania are unicellular protozoa, diploid organisms that have a nuclear genome of around 32 megabase and an extra nuclear DNA called the kinetoplast DNA, which is in part the equivalent of the eukaryotic mitochondrial DNA. So they cause a group of diseases collectively known as the leishmaniasis, which may have different clinical outcomes ranging from a self-healing skin lesion to visceralization affecting organs like the spleen, liver, and bone marrow, which can be fatal if left untreated. There's a strong correlation between the species of the infected Leishmania and the clinical manifestation. And here I'm showing just a few examples of that association. But we, in fact, know that there are more than 20 species known to infect humans. Leishmania are trypanosomatics with a digenetic life cycle, and inside the midgut of the phlebotomine sandfly vector, we find different forms of promastigotes, such as procyclics, which have a shorter flagellar length and multiply by binary fission. Procyclics represent just one type of promastigotes in the gut. There are at least 5 different types. So procyclics differentiate into nectomonads, which start a process of migration to the anterior parts of the gut. They then can become leptomonads, which then can become haptomonads and remain attached to the gut wall, or differentiate into metacyclics, which is a non-dividing, highly motile promastigote, which is the infective stage transmitted by the sandfly bite and is capable of differentiating into a mastigotes, which are these round-shaped forms that live inside the phagolysosomes of phagocytic cells, like neutrophils and macrophages. So during a new blood meal, sandflies ingest a mastigotes, and they can again differentiate into procyclics inside the sandfly midgut. Now, in the lab, we can mimic some of the conditions found inside the sandfly gut so that in logarithmic phase exinic culture, there's an enrichment for procyclic promastigotes. And in stationary phase culture, we find more of these metacyclics. But as to what extent that really reflects the in vivo conditions and the in vivo stages, it's not so clear yet. Right, so one of the most important questions about the life cycle of Leishmania and the sandfly is the cryptic sexual cycle. It is clear that genetic exchange happens inside the sandfly, but there are basic questions that still remain unanswered, such as we still do not have direct evidence of meiotic sex in situ, and most importantly, we have yet to identify the gametic stages. So could it be one of these different promastigote stages or an unidentified form, or maybe there just isn't one gametic stage in Leishmania? So those are the type of questions that we are investigating right now. So sex through meiosis has been described to have several advantages, such as to avoid the accumulation of potentially harmful mutations, and the genetic diversity that can be generated through meiosis. So in meiosis 1, homologous chromosomes are paired, and there's the possibility of resulting in recombination of the 2 different DNA molecules by a crossing over event. And so the haploid gametes that are generated may end up with different genetic content from each other and also from the parental cells. So after fusion of paternal and maternal haploid gametes, there is reassortment of different DNA resulting in more genetic diversity. So as a source of genetic diversity, meiotic sex has been extensively used in classical forward genetic study. So in reverse genetics, a gene is selected based on a hypothesis, and a targeted approach generate mutants, such as by gene deletion, and a phenotype is then studied. Now, in forward genetics, a phenotype is selected, genetic mutants are generated in an unbiased manner, using, for example, experimental crosses, and then the progeny or the mutants generated are screened for the specific phenotype, and then genes are associated or not with the phenotype. So the major advantage of forward genetics screens is its unbiased nature without the need of a hypothesis about the phenotype. So experimental mating in Leishmania could be a valuable tool actually for positional mapping of genes involved in host-parasite interactions. Sex is widely conserved in eukaryotes in general, and here I'm showing just a few different taxa where sex has been described. And these are highlighted by the bars in black. But in several protist lineages, there is still absence of evidence of sex. Although in Euglymozoa, which includes Leishmania and Trypanosoma brucei, for which meiotic sex has been formally demonstrated, we can see that sex is present and has been described. So an important evidence of sex is that in Euglymozoa carry homologs of genes involved in meiosis, as you can see here in more detail, where white circles represent the absence of that gene. So they do have genes involved in gamete fusion, double-strand break, homologous chromosome alignment, and recombination of DNA. So there has been long discussion on whether strict clonality would be the most likely model to explain Leishmania reproduction. But now it is widely accepted that genetic exchange can happen in Leishmania inside the sand fly, although the mechanism is still completely unknown. But perhaps the first report of fusion between Leishmania cells was observed by this group in Montpellier in France more than 30 years ago, between cells of Leishmania infantum and between cells of Leishmania tropica culture. But this, unfortunately, so far has not been reported by other groups, nor have we been able to observe this process in any way. So the first experimental demonstration of genetic exchange in Leishmania was described by the Sachs lab in 2009, showing that the process happens inside the sandfly gut. And from there, other studies were published by our group showing that an elevated phylogenetic conservation is not actually necessary for this genetic exchange to happen. And now we can use tools like whole genome sequencing to study the history of these crosses and have indirect evidence of meiotic-like recombination in Leishmania. In the lab, we routinely perform experimental co-infection of flies to generate hybrids. We just need two parental Leishmania strains carrying two different drug resistance markers. For example, neomycin resistance and hygromycin resistance. And then we co-infect the flies. And here's an example of a fly infected by RFP positive parasites, so expressing red fluorescent protein. Then we dissect the midguts from those flies and select for double drug resistant hybrids in culture. After sequencing the genomes of parental strains and the hybrids, we can compare each parent to a reference strain, which I'm not showing here, to find single nucleotide polymorphism, SNPs, and select for SNPs that are homozygous and different between the two parents. So this way we can track the parental homozygous SNPs as a proxy for allelic inheritance and therefore parental contribution in the hybrid genome. So in this example, homozygous SNPs in the Hig resistant parent are represented in red. and homozygous SNPs in the NIA-resistant parent are in green. And in this case, this chromosome is a disomic chromosome with 2 copies. Then in the genome of this hybrid, we see a parental contribution of 1-to-1, confirming that this is a full genomic hybrid. I mean, after we do a genome-wide analysis, not just looking at one single chromosome. So if we take this type of plot and do the same analysis for the whole genome, and then we fit all the chromosomes into a circle, we get this kind of plot, which is named circles plot, where each block between the white lines is a different chromosome from 1 to 36, and each track is a different Leishmania sample. So here we can see in orange homozygous SNPs for this parent, and in green for the other parent. And the F1 hybrid, so the first generation progeny hybrid, is showing full or near complete heterozygosity of those parental SNPs. And so a classic evidence of meiotic-like process is chromosome segregation and genetic recombination. And Leishmania F1 hybrids are actually capable of undergoing genetic exchange by backcrossing to one of the parents. And that leads to this kind of contribution of homozygous SNPs from the parents, where we can see several regions of partial or even complete loss of heterozygosity in many of these chromosomes. In reality, not many labs can use the sandfly co-infection model because they have no access to sandfly colonies. And sandfly rearing actually is not a simple task, which makes it hard for scientists aiming to study Leishmania genetic exchange. So a question that kept us awake for hours at night in the past few years was, can Leishmania have sex in standard lab culture medium? So that question was answered in 2020 by a report from our lab, where Isabelle Louradour developed a simple approach where she co-cultured 2 different parental strains carrying different drug resistance markers and fluorescent proteins, and she was able to select for hundreds of different Leishmania tropica hybrids in vitro. A key component of that system that made it possible to work really well was the choice of Leishmania strains, which are namely L747 and A-May37, both Leishmania tropica. And this specific mating pair has given some of the highest hybridization frequencies we have ever seen in the sandfly co-infection experiments. And yet, in vitro, the frequencies were much lower using this protocol. Other pair combinations have been reported recently in this communication using the same protocol, but at a much lower rate in other species, about 100-fold lower, showing that there is something specific about Leishmania tropica that make them highly mating competent. So Isabelle showed that we can screen for hybrids using flow cytometry really easily without the need of initial use of PCR. Here showing the RFP parent and the GFP parent and the double positive fluorescent hybrid. And the same was shown using microscopy analysis. It is important to mention, though, that even though the parental strains used are diploid, not all hybrids generated follow the same ploidy. So in this DNA content analysis here, using propidium iodide staining and flow cytometry, we know that the parental strains are diploid, but we can see that some of the hybrids generated are diploid, triploid, or even tetraploid. But in fact, this is not something new, as it can also be observed occasionally in some sandfly co-infections, as shown by our group in previous reports, although at a lower frequency than in the in vitro system. We can see here an L major cross where the parentals are diploid, and a few triploid hybrids were generated in this case. And also the same was seen in a cross between Leishmania major and Leishmania infantum in the sandflies, where an occasional triploid and tetraploid hybrid was generated. But just like the ones derived from the sandflies, the in vitro hybrids are full genomic hybrids, as confirmed by, again, tracking the inheritance of the parental homozygous SNPs from L747 and M837. And here shown in detail, we can see the one-to-one contribution of the parental homozygous SNPs in both diploid and tetraploid hybrids. But we can see that in the triploid hybrid, it looks more like a 2-to-1 contribution from the parental SNPs. So now we have this new reliable protocol to generate hybrids in vitro. The next thing we want to know is, can we use these super sexy Leishmania tropica to identify cells potentially involved in mating? And for that, we needed a technique to identify cell populations at very low frequencies. So we decided to use single-cell RNA-seq because of its use in other organisms to investigate cellular diversity and identify rare cell types in a systematic way. However, there were no reports of single-cell RNA-seq analysis in Leishmania at the time, so we did not know if this was going to work, but we decided to take the risk because it was worth it. In summary, one important difference between bulk RNA-seq and single-cell RNA-seq is that in the first one, you end up with the average expression of all the different cells in a complex sample, but in the second one, the transcriptomic information from each cell is preserved, and we can visualize the different clusters of cells after reducing the dimensionality and show the data as UMAP projection plots, where each dot represents a cell, and the distance between the dots tell you about the similarity in the gene expression profiles. And it also allows the study of the information in that way, even from less represented cells, which would be otherwise lost in the bulk analysis. We analyzed log phase culture of the two parental strains, L747 and M837, because that was the stage that gave the highest number of hybrids in the in vitro assay. And we used the variable genes identified as an input for the Surat R package to separate the cells in the different clusters based on the transcriptomic information. So this is what we found initially, looking at the UMAP plots of both L747 and M837. And what is very interesting when we compare the two strains is that they look very different in the UMAP plots and only partially clustered together. And here, just to highlight that each cell represents each dot represents a single cell. And even after identifying anchor cells and integrating the data, they can only be partially co-clustered in our analysis. And then in a split view of the data, we can see the 9 different clusters that were identified using our parameters with different transcriptomic profiles. And only a few clusters are actually common between the two strains, which are these ones highlighted here. So I decided to analyze the strains separately for most of this work to avoid any strain-specific bias here. Around 13,000 cells were sequenced for L747 and 8,000 cells for AMA37. Using our parameters, we were able to identify 5 clusters in L747 and 6 clusters in AMA37 according to their transcriptomic profiles. And the biological replicates in the experiment look very similar to each other, as shown here for L747 and AMA37. But it's important to keep in mind that the number of clusters is arbitrary in most single-cell RNA-seq analysis, and in this case, they do not correspond to the same groups between L747 and AMA37. So that cluster 1 here does not actually necessarily correspond to cluster 1 in this other strain. So although most promastigotes in this clonal log phase culture have very similar gene expression profile, there's still a lot of heterogeneity, and we can see from the expression profile of the top 1 marker of each cluster in each strain. So here shown for the 5 clusters in L747 and here for the 6 clusters in AMA37. Looking at previously described markers of 2 different promastigote stages, there's a clear segregation of cells expressing metacyclic and procyclic markers. And even though this is just a log phase culture, we can see a small fraction of the cells in AMA37 expressing the metacyclic marker and an even smaller cluster in L747. So it was interesting to see that 2 of the clusters of MA37 seem to be more segregated from the core of the promastigote populations. And the markers suggest that the expression of several stress granule markers in this cluster and ribosomal proteins, translation markers in this other cluster here. So getting to the main focus of the analysis, unfortunately, we could not find meiotic gene homologs highly expressed in any of the clusters that we identified, as represented here in the UMAP plots for 9 different meiotic genes. So to summarize the first part, in vitro mating between Leishmania tropica promastigotes can happen in standard culture medium. In vitro, Leishmania tropica hybrids are in fact full genomic hybrids with different ploidy levels. Single-cell RNA-seq of clonal Leishmania tropica revealed inherent single-cell transcriptomic heterogeneity in log phase culture. And this method can potentially be used to distinguish different promastigote stages within the same culture or even different strains. A second question was, is it possible to enhance even further Leishmania tropica mating capacity and increase the frequency of cells involved in genetic exchange by submitting the cultures to stress? And I say stress because different types of stress are often a trigger for differentiation between the Leishmania life cycle stages. Going back to a bit to meiosis, let's do a very quick comparison between mitosis and meiosis in eukaryotes in general. Taking one diploid organism as an example, the chromosomes are duplicated at the start of each process, so that each chromosome has 2 sister chromatids. In mitosis, the chromosomes align at the metaphase plate. Then the sister chromatids are segregated to the daughter cells, which are then also diploid. Now, in meiosis 1, there are several important steps, such as homologous chromosome pairing and recombination through crossing over. Then the homologous chromosomes are separated to the daughter cells of meiosis 1, which then enter meiosis 2, where the sister chromatids are then separated into 4 different haploid daughter cells. In Leishmania, there are many descriptions on the mitotic cell cycle, showing the segregation of the whole nucleus without the dismantling of the nuclear envelope. But we don't even know if meiosis is actually present in these parasites, so it's still largely unknown. One of the first steps that occurs in meiotic prophase 1 is the initiation of meiotic recombination, which is marked by the programmed double-strand break catalyzed by this enzyme SPO11. Then the extremities of the double-strand break are removed by exonucleases, and that recruits other proteins like VAD51 and DMC1 that will have a function in the repair of that break. So it was surprising to me that for decades, scientists have been using exogenous double-strand break agents like ionizing radiation and oxidating agents to induce the sexual cycle in organisms like Schizosaccharomyces. So we decided to do a similar approach in Leishmania. So Isabel used the in vitro mating assay, but then she first irradiated the parental strains before they were co-cultured in vitro, and that led to a modified protocol, which now works for different Leishmania species, not just Leishmania tropica, and because it increases the frequency of hybrid recovery by several folds. And we have shown that it works really well for Leishmania brasiliensis, infantum, and Leishmania donovani. We submitted early log phase promastical cultures to different DNA stress treatments, and we found that H2O2 MMS, which is methyl methane sulfonate, and gamma irradiation resulted in an increase of hybrid recovery using our in vitro protocol. Our estimation of minimum frequency of mating competent cells also showed a similar increase in the different treatment, different DNA damage treatments for both parental strains. By irradiating the parental lines with 6.5 gray of gamma radiation, we were able to generate dozens of F1 hybrids for different parental strains. For example, here I show the parental contributions in hybrids from a cross between two strains of Leishmania brasiliensis, one which is positive for the Leishmania RNA virus and another one which is negative. And so in the circus plot, we can see that there is parental contribution of both strains used here in these crosses, where green SNPs are represented in green. SNPs from the GFP positive parental line are shown in green, and SNPs from the RFP positive strain are shown in red. And we can see that the hybrids are near full genomic hybrids. The same can be seen here for a cross between a dermotropic Leishmania donovani from Sri Lanka and a visceralizing Leishmania donovani from India. And all the hybrids we have analyzed so far are full genomic hybrids, and there's contribution from both parental strains. However, even though these are F1 hybrids, we can see sites that show evidence of homologous recombination, which I'm highlighting here with these circles, where there is sudden change in the parental allele frequency in both crosses from Leishmania brasiliensis and also from Leishmania donovani, as seen here. So here's just a different visualization of the Leishmania donovani circus plots with these regions of partial or complete loss of heterozygosity highlighted in the F1 hybrids. And here we can see some cases where half of the chromosome show loss of heterozygosity, or even there is complete loss of heterozygosity in a chromosome. An interesting feature of these irradiation facilitated hybrids is that they are all polyploids. So far, we have never been able to rescue a diploid clone from these specific crosses. So this is a difference from what we got from the initial protocol with Leishmania tropica or with the sandflies. So here I'm showing a heat map of the different chromosome somi, so different copies of the different chromosomes from 1 to 36, where the light colors represent chromosomes with 2 copies and darker colors represent chromosomes with 3 or more copies. And in this first example, the parents are mostly diploid, and the hybrids are either mostly triploid or tetraploid. And we see the same in the other crosses we analyzed, where all hybrids are tetraploids, like this one, this cross for between Leishmania donovani strains, or between Leishmania infantum and Leishmania tropica. And except for this hybrid, which was shown to be mostly triploid, as well as this one I mentioned for Leishmania tropica. However, it seems like it's not the irradiation by itself that is inducing these changes in chromosome somi, because when we irradiated just one of the parental strains and analyzed both 4 different clones from that irradiated culture and the uncloned culture, we saw exactly the same profile, or a very similar profile as the initial parental strain. And here I'm showing as for comparison a diploid hybrid, a triploid, and a tetraploid Leishmania tropica hybrids generated using the initial protocol without the irradiation. But hybrids do not have 2 nuclei, as we can see here from DNA staining in this microscopy analysis. And also by transmission electron microscopy, here are the diploid parental lines, and we can see that the tetraploid hybrids analyzed all have 1 nucleus. So this means that they did go through both complete plasmogamy and karyogamy. To summarize the second part, DNA stress treatments greatly enhance the frequency of mating with different Leishmania species. Radiation-facilitated hybrids are polyploids, potentially a product of fusion of diploid cells, and suggestive of the absence of a potential trigger for reduction or meiotic division. Changes in parental SNP contribution suggests recombination breakpoints in F1 hybrids, possibly due to an incomplete meiotic-like event that takes place after diploid cell fusion. So another question is it possible to use this irradiation-induced mating process to identify promestical populations involved in genetic exchange in vitro? So we next compared the single-cell RNA-seq profiles of irradiated promastigotes with the untreated cultures, giving a total of 20,000 L747 cells and 16,000 M837 cells. And in this PCA analysis, we can see that the 2 strains are again very different from each other, and they actually responded differently to the irradiation. 7 clusters were identified this time in both untreated and irradiated L747, and the same for untreated M837 and irradiated, with some clear differences in the clusters identified between untreated versus irradiated in the 2 strains. Looking at the percentage of representation of the different clusters, it is clear that cluster 2 in L747 is expanded after irradiation, which can be seen here and here. And a similar expansion can be seen for cluster 1 in M837, which is clear in the stacked bars and also in the UMAP plot. But in this heat map, I'm highlighting the expression of the top 10 transcript markers in each of the clusters, where yellow represents upregulation of those genes in those cells compared to all the other cells in the data set. And here becomes evident the expression differences between L747 cells in cluster 2 between untreated and irradiated samples. And a similar difference is also detected in cluster 1 of M837 between untreated versus irradiated. Next, focusing only on these two clusters, we found by gene ontology enrichment analysis that a few genes involved in RNA binding and some kinases are differentially expressed in the two clusters induced by irradiation. But most importantly, the two clusters share a list of upregulated genes. And in that list, we can find some meiosis-associated genes. We looked at the expression levels of 9 different meiotic gene homologs, and we found that these are upregulated in the irradiated samples. So here we can see the darker blue representing the upregulation of the high expression of these genes, and the size of the circle representing the percentage of the cells that express those genes. And we can see that these 3 genes are upregulated, clearly upregulated in the irradiated samples. RAD51, which is involved in homologous recombination repair, GX1 in nuclear fusion, and HAP2 involved in gamete membrane fusion. And also we found this HAP2-like gene upregulated in these cells, but we still don't know what is the function of that gene. Looking at the single cell expression levels of HAP2 in UMAP plots, it is clear that the expression is concentrated in the radiation-induced cell clusters, which are cluster 2 here for L747 and cluster 1 here for M837. And a similar profile is seen for GX1 with a higher percentage of cells expressing GX1 in cluster 2 and in L747 and in cluster 1 in M837. HAP2 or GCS1 is actually an ancient gamete protein found in non-vertebrates in several different taxa, as shown here. And its structure is very similar to viral fusion proteins. So it's involved in the fusion of the membrane of the 2 gametic cells. Since HAP2 seems like an interesting marker for us to focus, I filtered for cells expressing HAP2 in the radiation-induced clusters in the 2 strains and compared their gene expression profiles with HAP2 negative cells in the rest of the data set. And we can see several genes being upregulated in these cells, like other meiotic genes, such as GX1, RAD51, and these HAP2-like, as well as a list of other genes that are not usually associated with genetic exchange, such as this gene with an unknown function, and a few RNA binding proteins like RBP5. Then to label cells expressing HAP2, we generated neon green HAP2 reporter constructs in both parental lines using Cas9. And then by flow cytometry analysis, it was clear that the different treatments increases the percentage of HAP2 positive cells in both L747 and M837, although at different rates between the strains. And we can see the same for irradiation, H2O2, and MMS treatment. We FACS sorted the M neon green positive and M neon green negative populations from both parents, and then we used them in our in vitro crossing assays. This resulted in a significant decrease in hybrid recovery when we used the two HAP2 negative populations compared with the two HAP positive populations. And we see a similar decrease when we looked at the estimated minimum frequency of mating competent cells. What is clear is that HAP2 is not an essential gene for promastigote survival in axemic culture, as we are able to knock out this gene using CRISPR-Cas9 in both L747 and A-MAY37. And when we use them, the null mutants in our in vitro mating assay, we see that it generates much lower frequency of hybrids when compared to when we use the two wild-type strains. And the estimated frequency of mating competent cells is also much lower. In summary, our single-cell RNA-seq analysis highlighted a discrete promastigote population with a transcriptomic signature that includes the expression of meiotic gene homologs. The HAP2 reported construct allowed us to select from mating competent and mating incompetent promastigotes. And finally, this new protocol removes the requirement for sandflies potentially to study Leishmania's sexual cycle and should facilitate the generation of recombinant parasites for positional cloning analysis. Finally, I would like to highlight here the three major methods used for experimental Leishmania hybridization. And you can see that sandfly co-infection still gives the highest estimated frequencies, around 10 to minus 4, while the initial in vitro hybrid method leads to around 10 to minus 8 when using L.tropica species, down to 10 to minus 10 for other species. But when we induce DNA stress before the in vitro co-culture, either by irradiation or a different DNA damage agent, we can reach 10 to minus 6. And down here, I'm listing all the successful Leishmania crosses reported so far for each of the different methods, either by us or different groups. With that, I would like to thank first the SBPZ for this amazing opportunity to discuss my work. And I would like to thank everyone from the SACS lab, which are wonderful people to work with. Particularly, I would like to thank Isabelle Louhadour, which is now at Institut Pasteur, and all the collaborators involved in this work. Also, all my other colleagues from the Laboratory of Parasitic Diseases at the NIH and TriTripDB for the genomics data resources. I'd like to thank you for listening, and I'm open for further discussions by email, which you can find down here.